Hey guys, hi. So this is the first question from the IBO 2013 paper. It talks about a special process of gene regulation, that is attenuation. We'll get to that in a while. But before we continue, this is what you must do. You must pause the video right now and try to attempt the question yourself and then come back to the video if you're familiar with the concept. If you're not, you can continue watching. I'll pause for 1 second here so that you can do that thing. Okay, let's get started. So some bacteria possess an additional mechanism to regulate the production of enzymes involved in tryptophan biosynthesis, the Trp Operon possessed prior to the actual genes trpA-E. So what is the operon concept? Let's talk a little bit about it. So actually they have given you here. So trpA-E, they are the, let me get the black color. So these are the structural genes. Now when I say structural genes, what it means is that these are the genes which are going to make enzymes, okay, and these enzymes at the end are going to synthesize the tryptophan, but we are clubbing them together. Why they are clubbed together as a unit here? So that they can be regulated as a unit, because we don't want to switch on one enzyme and not switch on other enzyme. That would be like having a loophole in the process. So we want these enzymes either to be switched on or off. By switching on, I mean the synthesis to be switched on and off. Regularly, we have an operator sequence, a sequence of the DNA within the promoter. So operators, they are nothing but DNA sequences. They could be within the promoter or outside the promoter, right? And far to the left, you have another which we call as a regulatory gene. Now usually tryptophan is a repressible operon. Now what is a repressible operon? The word is very clearly stating it out. An operon which can be repressed or which could be switched off. Usually it would be in a switched on state, but something switches it off. What does switch it off? Here comes the regulatory gene, which when transcribed and translated would produce a repressor. But unfortunately, this repressor is inactive on its own, but if it were to bind to a co-repressor, then it would be functional. Now you must understand, by default, this gene is switched on. The set of genes, they are switched on. So by default, the repressor is inactive and the tryptophan biosynthesis will continue to happen. Once we have a co-repressor, then opposite will happen. The co-repressor in this case is tryptophan itself. So this makes it functional. The regulatory product, now the active repressor will bind to the operator sequence and turn it off. Okay, so RNA polymerase cannot go ahead. Okay, now why did I go through this? So that you understand what kind of functionality happens within an operon. And another key fact, I said the regulatory gene makes the repressor after transcription and translation. So you must infer that in bacteria, what happens is many a times the process of transcription and translation occurs simultaneously. Now when I say simultaneously, they're not occurring at the same time. By this, it means that one part of the segment is being transcribed and the translation, it has begun a couple of nucleotides away, right? So at the same time, the part of DNA is being transcribed, and as the transcription proceeds ahead, a few nucleotides, actually hundreds maybe, the translation begins. Okay, so this was a regular what we just covered was a very regular form of control that happens. Now, attenuation is somehow a completely different story. Now, let us try to understand attenuation. Now, let's continue to read the question. So, at high tryptophan concentrations, the ribosome translates the mRNA of the leader peptide. Now, what's the leader peptide? So, let's focus on this particular sequence right here. So, this is a leader sequence, the DNA sequence. This will be transcribed into an mRNA and later on translated to a protein. Let's call this a leader peptide. Okay. The ribosome translates the mRNA of the leader peptide and stalls at its stop codon, thereby masking segment 2 of the mRNA and allowing segment 3 and 4 to form a stem loop. A stem loop is this U loop. A stem loop followed by a poly-U is a termination signal for the RNA polymerase which falls off the DNA. Now, at high tryptophan concentrations, logically, when we have a high tryptophan concentration, we do not want the synthesis to take place. Okay, so these are 4 segments of the leader sequence. So what happens is it can go through several states. So let's name them first. 1, 2, 3, 4. So this is 1. 1 and 2, 3 and 4 can form loops. 1 remains unlooped, 2 and 3 forms loops, and 4 again remains unlooped. Now, what will be the termination signal here is a 3-4 loop and a poly-U. If I have a 3-4 loop and poly-U, that would be my termination signal. I want that my operon should not do anything, should not transcribe and translate if I have enough tryptophan. Okay, so when I do have high levels of tryptophan, the tryptophan binds to a particular tRNA. Of course, you understand it's an amino acid. So amino acid carrying the tryptophan, I have this product tryptophan with the tRNA. If I have a lot of tryptophan, I'll have a lot of tryptophan tRNA complex, and if this complex is available, the ribosome, as it is translating the mRNA, once again I want you to remember the translation and transcription are happening hand in hand. So the RNA polymerase has barely reached this point, right? Just around 3rd and 4th part of the mRNA. Okay, the RNA polymerase has barely reached the point before the 3 and 4 part of the mRNA. Meanwhile, the ribosome has assembled at the mRNA near the 1 and the 2 points, and if I have plenty of tryptophan, the tryptophan tRNA complex will be there in enough quantities, and the ribosome moves to the 1st and 2nd segment. That means they cannot form a complex right now. Okay, they cannot form a complex. The 2 cannot form a complex. What does that leave us? 3 and 4. The 3rd and 4th do form a complex. We have a poly-U signal just after it. This will be a shut-off signal that I have enough tryptophan, let's not proceed. And the RNA polymerase falls off. Okay, now this is what they want us to understand. Okay, now let's look at the other scenario. However, low tryptophan conditions. If I have low tryptophan conditions, the ribosome, while translating the mRNA of the leader sequence, will stall, will wait for enough tryptophan tRNA complex. They will wait. So they'll get stalled at 1. Okay, they'll get stalled at 1. So when this 1 is busy, 2 and 3 can form a U loop. Okay, so 2 and 3 have formed a U loop, and if 2 and 3 form a loop, 3 and 4 cannot form a loop. I cannot have a termination signal. Okay, this should make very sense, because I don't want the tryptophan synthesis to work when I have plenty of tryptophan, but I want the tryptophan tryptophan synthesis to work when I do not have tryptophan. Okay, so if I do not have a termination signal, the RNA polymerase will continue, and very soon the ribosome would follow on the mRNA, and I'll have my tryptophan. Okay, so this is the concept of attenuation. Now we have the questions. So I hope this is readable. The first question is, the same regulation mechanism also works for the nuclear genes in the eukaryotes. Now here you are required to understand the difference between the transcription and translation in eukaryotes. In eukaryotes, transcription, that is going to happen inside the nucleus, right? That's going to happen inside the nucleus, and the mRNA must come out, and in the cytoplasm we have the translation happening. So there's no chance that this same mechanism will work. So I'll have to give it a false. Now, with reduced concentration of aminoacyl-tRNA synthetase, now what is aminoacyl-tRNA synthetase? Is, that means it's an enzyme. They have given you what it does. Attaches tryptophan to tRNA. Remember, our mechanism greatly hinged on tryptophan tRNA complex, because this is what the ribosome was reading or waiting for. If I have tryptophan, I'll have tryptophan tRNA complex, and my ribosome will not wait. Okay, it will not wait. 1 and 2 will be covered, 3 and 4 loop will be formed, my synthesis will be paused. So the question says, if there is less of this enzyme, transcription of the trpA-E genes will be inactivated. Now let's think about it. If I do not have the enzyme, irrespective of the tryptophan concentration, I do not have the enzyme, I do not have the complex. I do not have the complex, the ribosome will wait for it. Okay, the ribosome will wait for it. If it is waiting for it, right? If it's waiting for it, 1 will be busy, 2 and 3 will form a complex, and 3-4 complex will not be formed. Okay, that means the biosynthesis can proceed. It will not be inactivated. The synthesis will proceed. Okay, clear enough? Now, third question. After deletion of one of the 2 tryptophan codons in the gene coding for the leader peptide, transcription of trpA-E genes will be inactivated at a lower tryptophan concentration. Okay. The concentration of tryptophan is low. That means ideally what should happen? The ribosome should wait. Configuration 2. Okay. Ideally what should happen? The ribosome should wait at 1. Okay, but the question says you are going to delete. The question clearly states you are going to delete one of the 2 tryptophan codons, either 1 or 2. Now see, if I am deleting 1, so the ribosome will wait at 2. If it waits at 2, 3-4 is free to form a complex. Okay, so let's write it down. That's better, right? If I delete If I delete 1, what will happen? 2 will be busy. If 2 is busy, 3 plus 4 complex is formed, and this is a termination signal. If I do not have the 2, if the 2 is deleted, what will happen? 1 is busy, there is no 2. Once again, 3 plus 4 U loop is there, and this will lead to formation of a termination signal. So if either of one or the two is deleted, I will have a termination signal, right? So even if I have lower tryptophan concentration, there will be no transcription of A to E genes. Why? Because the termination signal will be formed. Okay, so transcription will be inactivated. That's a yes for us, right? That's a yes for us. In case of a mutation destabilizing the stem loop 2 and 3, mutation is destabilizing 2 and 3. Transcription of trpA-E genes will be inactivated at a lower tryptophan concentration. Now, usually, once again, lower tryptophan concentration means RNA polymerase should ideally, if I have a lower tryptophan concentration, I should get a 2,3, right? So that 3,4 is not formed. But the mutation is destabilizing the stem loop 2 and 3. Okay, what does that mean? That means 3 and 4 will be formed. If 3 and 4 will be formed, there will be a termination signal. And if there is termination signal, we will not have a transcription. Fair enough? So that means the D option is will be inactivated. That is true. Okay, let me use the green color. Okay, so I hope this makes sense, right? Some videos would be longer like this, some videos would be shorter, depends totally upon questions. And guys, if you're in India and if you're preparing for exams like NEET, there's a great book that I've just authored that's called NCERT Map. It allows you to make great connections. It has a lot of illustrations, a lot of diagrams, a lot of storytelling to help you prepare for your NEET or CUT examinations. Go check it out on the Amazon. The link is there in the description. You can totally get it. Take care. See you next time.